Thank you for joining our webinar today. I'll be your presenter, Brandon Muhlstein. I am a Packaging Study Director and Sales Liaison with Nelson Laboratories. Today we'll be talking about developing your packaging validation plan. And the reason why we are discussing this topic today is because we want to share several aspects that we have seen that have been successful over the past few years on accomplishing this apparent momentous task. For today, this is a brief summary of what we are going to be accomplishing by looking at standards, looking a little bit about the regulatory requirements, design, packaging, sterilization, protocols, and then briefly on testing. To start off with standards, ISO 11607-2 is the main standard that we will be mostly talking about today. It is the standard that describes and goes over in detail what is part of a packaging validation plan. TIR 22 is a great resource to help clarify and add some detail to 11607-2. ISO 16775 is actually a brand new standard that came out last year that is specific to the healthcare industry and reusables. ISO 11607-1 is the part of ISO that talks about the individual test methods and the details of the actual package validation. So why develop a package validation plan? One definition of validation is to recognize, establish, and illustrate the worthiness or legitimacy of. So by creating a validation plan, you're showing that your package system, your sterile barrier system, is worthy of maintaining its barrier properties until end of use. Also, it is to meet regulatory requirements and to show that, as we mentioned previously, that the sterile barrier is good until it is used. Regulatory requirements. Different medical devices require different requirements. Some require general controls, while others have more involved, such as special controls, while even others, and we'll discuss this in more detail shortly, require PMAs and 510K submissions, and even combinations of these. By understanding where your medical device and your process and your intent fits into the regulatory system, not only will you save time by not having to repeat testing later on down the road, but you can also lower overall sample and manufacturing costs and have a faster time to market due to these less complications. So moving on, prior to developing your plan, the plan actually starts months and weeks to months ahead of the actual writing of the protocol. It starts with the design qualification. The design qualification helps answer, why go down this road? Is it to fix a problem? Is it to make a process easier? Is it to be more efficient or even less invasive to a patient during an operation? Or is it a brand new idea that could revolutionize the market or a niche within the market? The DQ or design qualification examines that purpose of the validation and briefly describes what the intent and where the validation is going to go from here. Next step, as we talked about, is that under this design qualification, you need to know the path or the classification your device will go down. According to the US FDA, there are 3 classes that medical devices fall into: Class 1, 2, and 3. This table depicts each class by the type of device, the risk category, and the regulatory requirements. Let's go over Class 1 real quick. Class 1 is the lowest risk level, and Class 3 is the highest. But what determines the level of risk, or the requirements from a regulatory standpoint, or even how to classify the device or the types of devices within each class? Class 1 is classified as a low risk because the devices in Class 1 are not life-supporting or life-sustaining. Moving on to class number 2, these devices have both general and special controls. General control is things such as registration, enlisting, prohibition against adulterations, misbranding, notifications, repair, replace, refund, recalls, records, reports, and adherence to GMPs, which is good manufacturing practices. Special controls are post-market surveillance studies and performance standards. Class 3 devices are classified as high risk because they are life-supporting or life-sustaining devices. For example, in addition to the ones listed here, we have heart pumps and automated external defibrillators. So now that we have identified the appropriate class, let's start looking at the path. Most devices and ideas start with a computer program, usually a CAD design, where you start looking at the overall shapes, the sizes, functionalities, placement of buttons, knobs, levers, wheels, etc., but you also start looking at the other aspects that accompany the device, such as what materials the device will be made of, the packaging, not only in the type of packaging, but dimensions and materials, sterilization, the type, EO, radiation, steam, E-beam, etc. All of these things need to be taken into account early on in the phase, especially with medical devices. Keep in mind that different materials are more compatible with specific needs. Implants would be made more out of a titanium-based material, whereas a catheter would be more out of a urethane. So materials make all the difference. As we mentioned just previously, looking at different materials will help with later on testing and processes to make sure that things move as smoothly as possible. Some materials can only be sterilized by a specific method, so thereby selecting a material, you can actually find yourself cornered into a specific sterilization method, which in some cases may not work for your product, for your process, your intent, the operation, etc. And so you may need to look at readjusting what the materials are to get into a different sterilization so that it ends up working for you and your process. The next step, once we have designed the device using a CAD-based program, is to generate a prototype. The prototype can answer many questions, such as how does it actually function? Is the size appropriate for a surgeon's hands? Is the wheels in good reaching ergonomics to the rest of the device? Is it buttons, components? Is it easy to use? Prototypes are phenomenal for this because it gets it into your hands. However, it can also raise additional questions that you may not have thought of previously to having a prototype. A lot of this checking once a prototype is done is actually referred to as feasibility testing and can determine whether the design or the device is worth pursuing past this point. Depending on the device, you may find yourself needing several revisions of the prototype to make sure that the device and its configuration and orientation and size meets your end goal. It should be noted that your final device may not look anything like your original CAD design or even the first prototype, because those revisions are only ultimately making the device better for the end use. Prototypes also allow you to reach out to other companies, sponsors for funding or marketing to get it out there. They also allow potential clients to be involved in the development of the device by allowing that hands-on interaction to see how it works, see how it functions. Nurses and doctors who are in the field every day using these devices know what's going to work best in many situations and can provide valuable input on how the device should be developed correctly. So now we get into the plan. At a minimum, the plan must include equipment, IQ, OQ, PQ, software validations, quality system, and process interactions. Software validations and quality systems by themselves are enough to be their own webinars, so we'll only mention them in this one as areas to keep in mind and to look towards for additional information. Equipment. Sealers is the biggest portion when it comes to package validations and packaging your medical device. Depending on the needs, your application, the size, the end use, there are a variety of sealers, sizes, options, features, functions, etc. that can be purchased for your specific needs. Purchasing a good quality sealer and spending a little bit more time and research up front can significantly reduce issues, loss of time and samples because of insufficient or damaged seals caused by the sealers. Over the past year or two, we've seen several instances where low-quality sealers were used and implemented in the packaging system, and the packages themselves failed integrity and strength testing because the sealers that were used were unable to maintain a consistent seal across the entire package because of temperature, dwell, or pressure fluctuations, or the inability for the sealer to recover quickly enough to get to subsequent samples. We've seen that the first, half a dozen samples off the sealer were working phenomenally, but once you get going past that, because the machine couldn't recover and maintain the thresholds and the tolerances that it needed, the samples started to have issues during the sealing process. So, quality does make a huge difference when it comes to sealers. Keep in mind that once you have purchased a sealer, the sealer still needs to be validated, hence the IQ, OQ, and PQ. In some instances, portions of these validations may have already been completed. However, it's important to note that even though portions have been completed, it still needs to be captured and documented as part of your validation that these portions are complete and refer to these subsequent documents for those. The installation qualification, often referred to as IQ, looks at whether the sealer is level, whether it can be turned on, plugged into the appropriate power sources, looks at the temperature, the time, the pressure, making sure that those all are functioning properly. Looks at the software, if the sealer has that, to make sure that the software has been validated, that if it rotates or if it has a belt, that the timing of the rotation, the plates for sealing blisters or trays, all are consistent and congruent and uniform so that you don't run into issues when you move into production of them not working properly. The operation qualification, or OQ, looks at establishing the actual operating window of the sealer, establishing that nominal setting. Usually this is accomplished by looking at the minimums and the maximums of the sealer to help narrow in that operational window. Finally, once you've identified the window that the sealer needs to be operating in, you move into performance qualifications, or the PQ. The performance qualification is performed on 3 consecutive successful production runs at your nominal setting, and that these production runs are take into account multiple shifts, different days, changing of tooling if necessary, changing of raw materials, etc., just to make sure that the samples that are coming off the line are to the best they can be. There's an old phrase of using the correct tool for the correct job. Here, sealers are no different. By getting the proper and the most appropriate sealer for your process will continue to save you time, money, and energy. The next step is looking at the process interactions. During the feasibility testing that we've previously mentioned, we were looking at how the device was. As part of looking at the device, we need to look at how that product or the device interacts with the sterile barrier system and how that sterile barrier system will protect the device inside it. So here are some common packaging materials. There's actually three main types. We have sealable pouches, bags, and trays. We have sterilization wrap and rigid reusable containers. Keep in mind that depending on your product, your sterilization needs, the aseptic presentation of the product, the duration of the product being sterile, all come into account when selecting the appropriate packaging material. So, discussing each of these in a little bit more detail, sealable pouches, bags, and trays come in a variety of dimensions and materials. So not only selecting what type, sealable pouches, trays, or resealable rigid containers or wraps, you're also looking at, the type of packaging. Are you looking at a foil combination, or a Tyvek Mylar medical grade paper, poly? So those are all things to consider as well. Also, with the aseptic presentation, you've got heat sealable, you also have adhesive. So looking at what's going to be the best for your product, for your end use, and that aseptic presentation. Wraps are phenomenal for individual instruments or cassettes or trays. Things to remember with wraps is that they come in different sizes, grades, or weights of the material, and so by selecting the appropriate sizes, grades, or weights of the material, you help with making sure that you don't have extra bulk when it's wrapped. That can create air gaps and cause problems with sterilization, that it's not too light, that the tray is too heavy so you rip through it. All things that need to be taken into consideration with wraps. Reusable containers are great because they are designed to hold not only a single device, but also device and some of the equipment or tools required for an operation or procedure. A lot of these reusable containers incorporates filters either in the lid or the base that can help aid in air exchange for sterilization for EO or steam. Keep in mind that if you do have filters in those, you do need to make sure that the filter material that you are using does still provide that adequate sterile barrier to the contents inside. It's important that some packaging systems is better suited for certain products and sterilization, and so everything should be taken into consideration when deciding. Sterilization is the next logical step in the sequence because we've talked about the packaging, how it's used, and getting it to become that sterile barrier system. Obviously, there are multiple kinds of sterilization. I've listed 3 to 4 on this slide just for ease of the discussion for today. Steam, though it may be fantastic because it's fast, only a few hours to complete a steam cycle, it has extremely high heat, which may cause plastics to melt or to disintegrate to where you now lose that sterile barrier system integrity altogether. EO, on the other hand, is low temperature, so you don't have the problem with the degradation, but you have residuals and you have to deal with aeration following. You want to make sure that the samples are aerated appropriately so that they're safe to handle. Radiation, specifically gamma radiation, is awesome because it has complete penetration. Electron beam has limited penetration, but they don't have residuals and they have lower temperature. But on the flip side, they do have material degradation because you're irradiating the samples. Protocols. There's actually two main types of protocols that we've seen. The first one being the validation protocol and the second being the testing. Validation can also be referred to as the master protocol. This validation protocol, master, describes the samples, the lot numbers, the size, the intent. It also provides rationalization and justification for the sterilization process and parameters that will be used, test methods for your biocompatibility, your packaging, list sample sizes for those different tests, and also includes your sample acceptance criteria, what determines that the results passed or failed. It also includes references to the testing protocols or procedures out there. The second type of protocol is the testing protocol. The testing protocol describes how the tests are performed specifically, the detailed step-by-step process. It also includes whether the test methods have been validated and if they've established sensitivity or precision and bias if it's applicable to the test methods. These testing protocols also provide the test method acceptance criteria, which would be how do you know that the results obtained from the test are valid? Not that the results are good or bad, but that the results are valid. Continuing on with protocols, we've seen several main components, and these ones are just a sample of the common components that we've seen in the validation or the master protocol. Keep in mind that these are just the common ones. You may find that depending on your situation or circumstance or device, you may need to add additional ones or remove as necessary. These protocols, these validation protocols, can reference other protocols like those individual test method protocols. By referencing those smaller test method protocols, updates can be made to those without jeopardizing your validation or master protocol. Worst case. Throughout ISO 11607, both Parts 1 and Part 2, worst case is mentioned several different times. Each time that it's mentioned, it's actually referring to different things. Two of the times worst case is mentioned, it is related to the device and the material. Here, with the device, we are looking at identifying the worst case device, the heaviest, the one with the most connections, the one that's the bulkiest, the one that has the most stress points on the packaging. It's looking at the device and selecting the worst case device. The second one that I mentioned was the material, looking at your sterilization process, making sure that you've got the validated temperatures, the pressures, the dwell times, multiple sterilization exposures. With EO, that's usually 2x on a full validated production cycle. With radiation, you're looking at 40 plus kilogray. The third time worst case is mentioned throughout ISO 11607 is actually referring to the manufacturing process, or the IQ, OQ, PQ that we've previously discussed. Don't forget to justify why a certain device or package system or sterilization is the worst case. This needs to be documented and justified so that it's captured correctly. ISO 11607-1 is the standard for helping to actually perform the package validation itself. 11607-1 says that you need to address 3 different categories: strength, integrity, and microbial barrier. Within each of these categories, there are several options that are available to select from depending on the material combinations, sterilization, and your end use for the sterile barrier system. Here we'll go over each one of these categories independently. For the strength category, we have 3 options: sill pill, burst, and creep. Sill pill looks at how much force is required to peel the sill apart. This value helps with making sure that you have a strong enough seal to maintain the sterile barrier, but not strong enough that you lose your aseptic techniques and ability to open it in an operating environment. The burst test is a whole package strength test that looks for the maximum pressure the package or sterile barrier system can withstand before it ruptures open. The creep test is excellent for non-porous type packages because you are subjecting the package system to a lower pressure for a duration of time to see if the seals creep open. For the integrity, we have 4 options: dye migration, dye immersion, bubble emission, and visual inspection. Dye migration is specifically looking for channels through the seals to indicate a breach in the sterile barrier system. Dye immersion is specific for rigid containers and looks at the closure system to make sure that they are intact. These are usually for drug type vial configurations. The bubble emission test, unlike the dye migration test, is a whole package test where you're looking at the integrity of the entire package, the seals, as well as the materials for any sort of gross defects. Visual inspection is exactly what it sounds like. It's a visual inspection of the packaging for channels through the seals. The standard does give you options that you can inspect for that may be applicable to your situation if necessary. The last category, microbial barrier, here again we have 4 options: the F1608, microbial challenge, F2638, and air permeance. The ASTM F1608 or microbial ranking is specifically to porous materials only, and it determines how well your porous material is able to prevent microorganisms from passing through it. The microbial challenge test is a whole package test that also uses microorganisms, but it's challenging the entire package, not just the porous material. The F2638 test is also a porous material like the F1608, but instead of using microorganisms, it uses latex spheres and variable flows to find the penetration max % of the porous material. Air permeance looks to see how well your material is non-porous. This one's specific for a non-porous type of packages. So in order to understand which of the tests we'd recommend and would be most appropriate for your device and situation, we first start out by looking at the sterilization method. Then from there we look at the material combinations, and then we get some suggestions. So let's say, for example, we're using ethylene oxide as our sterilization method, and we have a Tyvek mylar pouch. We would recommend a bubble emission test to meet the integrity category of ISO 11607-1. We would recommend a seal strength for the strength and the microbial ranking, or ASTM F1608, for microbial barrier. One question that we see quite often is, well, what if I have this historical validation and in it we have dye migration instead of bubble emission? Can I use the dye migration test instead of the bubble emission test? And the answer is obviously yes. The recommendations that we have listed here are what we have seen from successful FDA submissions. Obviously, if you've got procedures or historical data or other things to indicate a different test than what we have recommended, we can obviously make those adjustments for you. Also, along those same lines, you may find that your company already has specific procedures in place that dictates what test methods to select from, or risk assessments that have been done on different test methods that are appropriate for your company's needs. Also, historical data or preferences from quality assurance or other regulatory bodies looking for specific tests. In these situations, you should comply with these other procedures that you are faced up against. So what's next? Now that we have a written protocol, what are the next steps? Next is usually the sterilization validation. Sterilization validation will depend obviously on what sterilization method you're using. The different methods have different techniques to accomplish that validation. So for this webinar, we won't be going into the details of individual ones, but just mentioning to follow the requirements outlined for your specific sterilization method. Once the sterilization has been completed, most medical devices will go through some basic biocompatibility testing to show that the materials selected for the device do not have an adverse effect. Though the list of required biocompatibility tests will fluctuate depending on the device and its use, cytotoxicity, sensitization, and irritation are the three basic tests that every medical device has to go through that come in contact with the patient. In many situations, the biocompatibility and the package validation portions can be performed concurrently, which will help reduce the overall project time to market. Sterilization validation is done first because you want to make sure you're using validated sterilization parameters for your packaging biocompatibility testing. So once all the testing has been completed, the data needs to be reviewed and determine whether or not the results passed. This is usually accomplished by compiling the data from all the different phases, the sterilization, biocomp, the package validation, and comparing the results to the acceptance criteria, the sample acceptance criteria listed in your validation or master protocol. Assuming that the data meets those acceptance criteria, the next step would be to file with the FDA and go to market. Do keep in mind that any issues that arise at any of these different phases can bring your entire project to a halt. It can even make it go back to the drawing board or could even scrap the entire project altogether. So another question we get quite often is about revalidation. ISO 11607-2 gives a great section on what can qualify to be revalidated. Some of these changes are changes to the sterile barrier system, which could be a change in dimensions of a sterile barrier system, changing of materials, changing vendors of the raw materials. Another one could be process equipment, a brand new sealer, changing the sealer from one location to another, completely changing your processing plants, changing the device, changing the materials or the way the buttons or levers or things are situated could rub differently on the package system and could cause micro holes or holes in the packaging, which could cause other issues. Sterilization could be a huge one. This is one reason why we recommend running your package validation after the sterilization validation is complete, so that you make sure that the sterilization you're using has been validated. End user complaints or negative trends in quality, maybe changing of the transportation system, going from a local delivery process to a larger scope. You're used to sending out one box of 10 units and now you're changing it to sending out a pallet. All those things can cause a revalidation to be necessary. It is important to note that some minor changes may not require a revalidation, but it needs to be documented and justified in that the revalidation is not required. Also, you should look periodically at your minor changes, because one or two minor changes may not require a revalidation, but 5 or 6 minor changes may qualify it to be needing a new revalidation. Keep in mind that revalidation may not be a full repeat of what was previously done, but looking at showing equivalencies and looking at smaller, refined validations. So when you are looking at selecting a test lab, it is important to look at their accreditations. Nelson Labs is A-class accredited. We are also ISO 17025 accredited, and we have several members of our staff that are ISTA technician certified for the transportation distribution type testings. We also just successfully completed a TGA audit for those who are in our Australia locations that are looking for testing. It is also important to note that Nelson Labs has numerous staff members that lead and participate on the technical standard committees and methods, from ASTM to ISOs to ANSIs and AMIs, all across the board. We have members that participate on those very frequently. We believe that because every test matters, we win when our clients win. And so we strive for continuous improvements throughout every aspect of our company, from sample receipt to testing to reporting to review to sample return, trying to show and make improvements where necessary to help you as a client succeed and win. We try to stay at the forefront of the industry by improving the level of testing excellence and quality, not only here at Nelson Labs, but by participating in those committees across the industry. This brings us to the end of the webinar. I appreciate and thank you for your time. If you have comments or questions, please leave those, and we will answer those as they come in. Again, thank you for your time, and we will adjourn this webinar for today.